Today, we are not going to learn biology, but we are going to go on a mission to steal a biological code in the form of a gene, and then we will give this code to our secret agent who will take it, sit on a car, go and deliver it to our customer, and our customer, after getting the secret code, will use the secret code and produce the given product on a large scale and make it available for the public. Today, we are going to create something called as recombinant DNA or rDNA. So we are going to steal the secret code. In this case, the secret code or biological code is nothing but the gene. We are calling it as our gene of interest. I hope this is clear to you. This is the gene of interest that we are looking for. Now, where we will find this gene of interest? Let's say we are going to steal the gene responsible for producing insulin, human insulin. So, where we are going to get it from? Definitely from DNA. But we don't need the entire DNA. We have to isolate the DNA first from the organism, from the desired organism, in this case, a human being. But we don't need the entire DNA. So first of all, we are going to isolate the DNA. And from that DNA, we are going to specifically isolate that particular gene which is responsible for producing the insulin. Insulin is nothing but a protein. It acts like a hormone. So how we are going to do that? Using a restriction enzyme, which we also call as molecular seizure. So using that molecular seizure, first of all, we are isolating the DNA. And then from that particular DNA, we will cut out only that portion, the gene which is responsible for producing the human insulin. Then we are going to put it inside the plasmid. Now what is plasmid? Plasmid is acting as the agent over here. We are going to cut the plasmid using the same restriction enzyme, the restriction enzyme that we used for cutting the gene from the DNA, the same restriction enzyme we are going to use for cutting this particular plasmid. Plasmid, as I said, is going to act like an agent. So after cutting this particular plasmid, I am going to insert my gene of interest in this particular plasmid. Let me give a small background of this agent called as plasmid. Plasmid is nothing but it is a small circular double-stranded DNA, extrachromosomal DNA that is present, that is normally found in prokaryotic cells like bacteria. Now, what is the purpose of plasmid? We don't have plasmid, but we are we consider ourselves as very complex, very diverse. We don't have plasmids, we don't need plasmid. As I said that they are small circular extrachromosomal DNA. That means they are not part of the chromosome, they are extrachromosomal DNA, they can replicate on their own. Plasmids are usually found in bacteria because plasmids, they provide bacteria with certain unique features. For example, plasmids, they have certain antibiotic resistance gene. That means if the bacteria takes up this plasmid that contains the antibiotic resistance gene, the bacteria will get resistance from the antibiotic. That means even if we grow the bacteria in a culture that contains, suppose, ampicillin or tetracycline, even if I grow the bacteria in that media which contains this kind of antibiotics, the bacteria can grow easily because it acquired a resistance against these kind of antibiotics because of the antibiotic resistance gene which is given to it by our agent plasmid. So this is the gene of interest. This was the secret biological code that we are looking for. I have inserted this into the plasmid. And as I said that I will cut the gene of interest and the plasmid. I will cut the gene of interest and the plasmid using the same restriction enzyme. Using the same restriction enzyme, I am going to cut the gene and the plasmid. So it's like you go to collect the secret code, and then you have to show certain icons that I'm coming from this boss, give me the secret code, give this, I'm the agent, so give me the secret code. So it's like that only. Okay, so this is the plasmid, the secret agent. This is our biological code. I will cut these two with the same restriction enzyme. I will insert it like this, and thus my recombinant DNA, rDNA, it is already created. Very simple. Now I will put it inside the host. In this case, the host is generally, if we go for human insulin production, the host is generally E. coli or Escherichia coli, which is a very common bacteria which is used in a lot of biological experiments as a model organism. So when I am going to put this recombinant DNA or rDNA, how will I put it inside the host? Let's say bacteria. There are various methods by which I can do. One method is heat shock. Heat shock or calcium chloride treatment. This is a technique by which I change the permeability of the cell membrane. Bacteria, as we know, they have a cell membrane. So this cell membrane's permeability I will have to change, so that if I change the permeability of the cell membrane using the calcium chloride or by using the heat shock method, then what is going to happen? The permeability is going to change and the bacteria is going to take up the plasmid. That is a way by I can do it. Another way by which I can do is by electroporation. I am going to give small electric shock, as a result of which it is going to create certain small pores in the bacterial membrane, and through these pores the recombinant DNA will enter. Now, how will I ensure that the bacteria, it has taken up the plasmid? How will I ensure it? There comes a process called as selection. So suppose my plasmid, as I said, that plasmid provides certain special features to bacteria, for example, antibiotic resistance. So I will have to look for that what kind of antibiotic resistance gene is present in this plasmid. Suppose it is ampicillin, for instance. So I will have to create a plate, agar plate, that contains this ampicillin, and then I will grow it. I will just spread the bacteria. I will grow the bacteria, I will spread over it. Only those bacteria which has taken up the recombinant DNA successfully, only those bacteria will survive, and the bacteria without the plasmid will eventually die. So that is how we can perform the selection procedure. So let me summarize. First, I will have to isolate the DNA from the desired organism, the organism from which I want to isolate the DNA. For example, if I want to go for human insulin production, I will have to take it from human beings. So isolate the DNA from the human beings, that is the first step. Second, I will also isolate a plasmid, and plasmid we are not going to find in any sort of in any sort of eukaryotic cell. I will have to go for prokaryotic cells like bacteria to isolate the plasmid. Plasmid isolation is a very common step that we perform. So I have isolated the plasmid and I have isolated the DNA, right? But I don't need the entire DNA. I just need a small portion of it, right? I need a very small portion of it. This is my gene of interest. The entire DNA is not responsible for producing human insulin, that protein. Only a very small portion I need. This much, maybe this much. The DNA is this much. But I need a very small portion, this gene, which is responsible for producing the human insulin. So this is my gene of interest. How I am going to cut it? By using a molecular Caesar or restriction enzyme. Then, and remember, the restriction enzyme that we are using for cutting this gene of interest, it should be same. The restriction enzyme that I am using for cutting the gene of interest, the same restriction enzyme I will use for my plasmid. Then I will insert it and I will have to join it. The joining work, as you know, is done by the DNA ligase enzyme. So my recombinant DNA is prepared. This recombinant DNA is absolutely prepared. So this is my agent or you can call it as a vehicle also. And this recombinant DNA, now it has to be inserted. As I said in the story, that first I'm stealing it, I'm stealing the biological code, I'm giving it to my agent, and this recombinant DNA is like a vehicle. Now it has to be delivered to my customer. Here, the customer is actually the host, E. coli. The customer will use the secret information that is stored inside this gene to create a valuable product, for example, insulin, on a very large scale, and then we are going to sell it in the market. So this recombinant DNA, it's a wonder in biological field. So many applications are there. As I said, human insulin, Eli Lilly company, artificially creating insulin, which is very, very essential for the diabetes patient. So creating that artificially itself is a remarkable feature. It's a remarkable feat in the history of biotech. So that is how you create recombinant DNA. There are certain fireflies or certain jellyfishes are there, they glow. So the glowing jellyfishes is nothing but a wonder, or it's an application of this recombinant DNA. So creating the recombinant DNA is very important. And I also mentioned you about how we can transfer it. There is a process called as transfection by which we can do that. That is heat shock method, calcium chloride method, electroporation method. All these methods we can use for transporting it into the host, right? We can easily transport it into the host. And then the host will multiply and the plasmid will also get multiplied by that method. And then we go for the selection method. Only those cells which take up the plasmid, they will be able to survive in the plate. When we are plating it, only those that have taken up the plasmid, that will be able to survive because they have that antibiotic resistance gene. But those cells without the plasmid will not have that. They will die. And thus, we can select only those bacterial cells, those who have taken up the plasmid successfully. And thus, we have created recombinant DNA, we have introduced into the host cell, and we have selected only those bacterial cells that contain our recombinant DNA. And as the bacterial cell containing the recombinant DNA will multiply, because they have to grow, they have to divide, they have to reproduce. And in case of prokaryotic cell, as we know that reproduction is similar to that of dividing, they just grows by binary fission. So as they are growing, we are also getting our product, that is insulin in this case. So it has a huge application and it can be used for producing so many valuable products for the benefit of the human.